Good day, students. Welcome to the continuation of the theory related to mineral determination. In this section, I'm just going to focus on the different methods that are used to quantify the different types of minerals in food. Just a little bit of recapping. In the previous recording, we spoke about ways of analyzing or determining minerals in food. And then we said the simplest way or the simplest procedure that is done is etching, whereby we just determine the crude mineral. So we just talk about the total minerals in that food sample without actually going down to identifying a specific mineral or its concentration. I mean, in food science and technology, if we say minerals are important because they provide functionality in terms of the product, we mention something like solubilizations of proteins. We mention things like your preservative functions. And then we said on the nutrition side, obviously, it helps in terms of curbing deficiencies. Like we mentioned iodine that prevents goiter. So it means that if we want to be able to perform all those functions, we need to be able to go a step further, not just determining the total minerals, but quantifying and specifying which minerals and how much it is in there, so that we can be able to know whether we need to fortify it so that we can overcome the deficiencies. So of the methods that we had mentioned were three methods that I'm going to dwell on now. The first one being your titration methods, and then the second one will be your spectrophotometric methods, whereby we rely on color formation, and then the third one will be your ion selective electrode that works in the bases or similar bases as your pH meter. So those methods can actually do the identification and also quantifying the amount of those minerals in food products. So like we already mentioned, the precipitation titration methods is used when at least one of the product of the titration is an insoluble precipitate. So in that way, an insoluble precipitate is formed that can then be titrated or with another chemical to then identify or measure the amount of the mineral in there. And we normally use this for determination of chloride, or we can say salts or chloride in food products. Based on this, we then have two major methods that are your precipitation reaction methods. We have what we call the Mohr's method and then the Vollhardt's method of determination. Very important when looking at different methods, especially methods that can measure almost the same thing, like it's a duplication that Mohr can also do the salt and Vollhardt can do that. What we actually look for will then be the specificity in terms of the principles. Are the principles similar? One, know the principle. Number two, know the exact procedure, and then other environmental conditions that are related to that specific method. Know the advantages of one over the other. And then important, very important, you need to know for what type of food product is that method applied. Is it for all different food products, or is it for solid product? Is it for raw material, or is it for liquid product? So if you know all those four aspects, then you can actually master any method in food analysis. So the first method is the Mohr's method. The Mohr's method is the easiest and most straightforward method, and that's why because it uses forward or direct titration. So in terms of the Mohr's method, we just need three things. The first one will definitely be your sample that requires to be analyzed, and then secondly, we need a known concentration of silver nitrate, and then the third one, we need an indicator solution, which is potassium chromate. So what actually happens in this method is, when looking at the diagram in there, this method then determines the chloride ion concentration in a solution by titration with the silver nitrate. So when the silver nitrate solution is slowly added into the sample, a precipitate of silver chloride actually forms, because then that known concentration of silver nitrate that is added in excess will then complex all the chloride. When all the chloride is actually complexed or precipitated, the excess, the leftover, the silver that did not react then, the slight amount that did not react then, reacts with the potassium chromate, indicator, and then it forms a red brownish precipitate of silver chromate. So usually whilst you are still titrating and the silver nitrate is complexing the chloride, the color of that solution is usually cloudy white. But then when the end point is reached, it becomes a red-brown precipitate of silver chromate. So this method, very easy and straightforward. It can be used to determine chloride ions concentration in water samples from any sources, whether it can be your seawater, your river water, and so on. This method is more suited for solutions. And the reason why, we already know that your salt, especially your sodium chloride, then dissociates in water and forms sodium ions and chloride ions. So they are already in simple forms. There's no step that's required to do the extraction, so it becomes easier. Number one. Number two, your Mohr's method is actually used whereby the pH of the solution that's being titrated is usually around the alkaline pH between 6.5 and 9, and some textbooks will also tell you 10. So that will be the limitation or the conditions that are required for the Mohr. The one disadvantage will then be, obviously you can't use it for a product that is complex like your meat, because remember, there you need to extract that sodium chloride that is already attached to the proteins there, forming bonds and whatsoever. And then thirdly, the one disadvantage about the Mohr's reagent or the Mohr's method is that in terms of specificity, it is supposed to analyze for sodium chloride. But if your sample contains bromide ions, it also quantifies those bromide ions. So meaning that the silver nitrate can also, or the silver can also complex the bromide ions, and then in that way you can actually overestimate the chloride. So it won't be all sodium chloride, there will also be bromide in there. So that will be the drawback. So look at table 21.3 in Nielsen and know this exactly the chemical reaction that takes place. Okay, and then secondly, when we go to the Vollhardt method, the Vollhardt method is quite complicated, as I already mentioned. It's complicated in terms of the procedures that are used, and also at the same time, it's complicated in terms of the chemicals that are required. As I had mentioned, in the Mohr, you had two chemicals. You had the potassium chromate indicator and then the silver nitrate. However, when we do the Vollhardt method, that one has got more chemicals. First of all, surely, because you are analyzing for a sample, you will need a sample, number one. And then number two, you will then need silver nitrate. The silver nitrate is still required. You also need nitric acid. You need your nitrobenzene, and then the potassium thiocyanate. So you can see that it already has more chemicals compared to the Mohr's reagent. And then also part of the procedure that you will actually learn about when you do your practical is that there's a boiling step that's required. And that boiling step helps in terms of separation of the chloride ions that are attached in food products, whereby they are performing specific functions, especially your proteins. And then where it's also bound to proteins to actually bind more water. We'll talk about that when we talk about proteins. So then it becomes complicated that you need to remove that analyte, separate it. We had mentioned that in terms of sample preparation. So then what actually happens in the actual process? So as I had mentioned, this is a complex procedure. This method actually uses a back titration with potassium thiocyanate to determine the concentration of the chloride ions in solution because it's been separated. So the steps here would be, you have your sample that is in liquid form, and then what happens is that you add your excess volume of silver nitrate in there, and then you boil it, and in that way the silver nitrate there will then obviously react with the sodium chloride. So it happens in this way, whereby the silver still does the same thing as the Mohr's reagent. So in terms of the principle, it follows more or less the same principle. It's just that this one has a further step, whereby the silver will then react or complex all the available chlorine, because we mentioned that this is formed, is added, the silver nitrate is added in excess of the chloride ions. So when all the chloride ion is complexed, it forms what we call the precipitate, a silver chloride precipitate. And then what happens is that in this reaction, something that I forgot to mention is that instead of the potassium chromate indicator, we actually have a ferric ion indicator. So the indicators are also different. So from there, when the excess silver that is there that did not complex with the chlorine, because all the chlorine ions has been actually chloride ions has been complexed, that excess silver then reacts with the potassium thiocyanate to form silver thiocyanate. And then this is then used to quantify the silver that was not obviously complexed with the chloride. So the silver that did not react with the chloride, the excess, then reacts with the what? With the potassium thiocyanate, forming silver thiocyanate. And then what happens is that that other thiocyanate that is excess to the silver, because it's the back titration, then reacts with the ferric ion indicator, and then it forms a red precipitate. So that red precipitate forms when there is any excess of the thiocyanate that was not complex to the silver. This might be a bit complicating, but when you go and read it on Nielsen, you will actually understand. So I'll just repeat this again. So what happens is that the first step is whereby you have an excess volume of your silver nitrate that reacts with the chloride, and it complexes all the chloride. However, because it's excess, the leftover silver then reacts with the potassium thiocyanate to form silver thiocyanate. However, not all the thiocyanate is going to react. So the leftover thiocyanate, that's the one then that reacts, it's a thiocyanate ion that reacts then with the ferric indicator to form a red precipitate. So when the slight color change to red, that's already showing you the excess thiocyanate. And in that way, once all the silver ions have reacted, the slight excess, as I already mentioned, of thiocyanate reacts with the iron to form a dark red complex, and that will be your end point titration. So this concentration of the ion is then determined by subtracting the titration findings of the moles of silver ions that reacted in the first place, and with the moles of the silver nitrate that was actually added in the solution. Very important differences besides the differences in terms of the complexity of the Vollhardt method. The Vollhardt method is actually applicable whereby the pH of the medium is actually acidic. Let's say maybe you are titrating looking for sodium chloride maybe in beverages that you know that are in high in acid, or yes, that are in acid high acid levels. So then you use that. However, if you were to measure the same using the Mohr's method, it's not going to function because Mohr requires higher pH. And then also, like I already mentioned, that there are other chemicals that are added in there, is that the nitric acid that is actually added there, it is actually added to prevent the hydrolysis of the iron. So if the iron becomes hydrolyzed, then we actually have a problem in terms of the end point. And then we also have the addition of your nitrobenzene. The nitrobenzene is actually added to remove the silver chloride because it masks it. By masking it, it doesn't want it to then not like, you know, when a precipitate forms, sometimes it happens that when there's the excess of the other chemical that was previously associated with one chemical that forms the precipitate, it then reacts with that chemical again. So to prevent the reaction of your silver with your nitrates, then you mask the silver chloride using nitrobenzene. So it actually becomes a complicated reaction. So that is why it requires more steps and careful selection of the chemicals that are actually required. Like I already mentioned, that in terms of your products, with your Vollhardt's method, it's well suited for products with high salt, and also it can actually analyze a range of different food products, especially when we talk about complex products like your meat products, because there's the boiling step that is required to extract the analyte, which is your sodium chloride from the matrix of the food. And then obviously, just like any other method in food analysis, there's some calculation that has to be done, because remember, all the chemicals that are required there, we know their concentration and we know how much of that is added in terms of volume. And then also when titrating, we check the meniscus as to how much of that solution reacted. Okay, so you will do this practical on the virtual practical on Blackboard, so then you will actually understand it much better. Okay, and then the second one will be your colorimetric methods, whereby we use a spectrophotometer. So the principle here is based on, in this method, we have a chromogen. What a chromogen is actually a chemical that will then react with a certain compound to form a colored product. Okay, so in this principle, the chromogen will then react with a specific mineral. Remember, we're looking for minerals. So this specific chromogen will react with a specific mineral. So meaning each and every mineral will then have its own specific chromogen, and something that research has already done to know which one will react with what. And then when that chromogen reacts with the specific mineral that you are looking for in the food, then that product that is formed there will absorb at a specific wavelength of light, and that can also be then determined on or in a spectrophotometer. Okay, so the relationship between the absorbance and the concentration of your analyte will be given by the Beer-Lambert's law of A is equals to ABC, where A is the absorbance of obviously the product that is formed, the absorptivity, how the light passes through, the path length through the solution, and then also the concentration of the absorbing species. So in that way, you can actually construct a standard curve, and you can be able to read off the concentration of your analyte or your mineral based on the absorbance of the samples. And then, very important in terms of all these methods, we mentioned that the samples should generally be etched and solubilized, because remember, you can't just take your food sample and then you just add that chromogen. Remember that chromogen must look for the analyte. But what if the analyte is actually lodged within the food matrix? So there is some form of pre-preparation steps that you need to do. So the samples will then have to be etched, it has to be solubilized, and possibly it has to be treated via oxidation reduction to ensure that the mineral is in its correct form to be able to react with the chromogen, because now you can say the ion has to be in its reduced form and not its oxidized form for it to be able to react with that chromogen. And then when that happens, when the analyte is quite prepared, the reaction will be rapid, and then the endpoint will be stable, because the analyte is in a form that is required. And then when doing this, in terms of quantification using your spectrophotometric colorimetric methods, the time set in the experimental protocol for the color development can be seen. So what happens is that you look at the reactivity, you take the chromogen, it reacts with the already prepared analyte, and then over time you check the color formation. Or it can just be an instant thing. So and then you can actually monitor the formation of the color over time, and that will be the basis of analysis. So this is used for determination of phosphorus. So phosphorus is usually determined using your colorimetric methods. And then we also have the ion selective electrodes, where I told you that this works in a similar principle as your pH electrode. What happens is that, as I already mentioned, through research, a whole lot of things have been researched already, whereby they know that a specific electrode will work for a specific mineral. So each and every time when you want to analyze for that mineral, then you will use that specific electrode, because it can actually detect that mineral based on the configuration of chemicals that are in there that are actually sensitive towards that mineral. So the specific electrode is then used for a specific mineral, which is attached obviously to a pH meter, and then the reading of the determination of the ions concentration is used by measuring the electrical potential. So when that analyte passes through, its electrical potential is then measured, and then it will then be able to identify, oh, this is iron, this is chlorine, or so. So there's different types of electrodes that are used. They are normally used to measure chlorine or chloride. Therefore, then you can actually substitute the Vollhardt method, because that one is quite tedious, with the ion selective electrode. You can use it to measure your calcium, your bromine, your potassium, and your sodium. So it's quite easy to use and much faster. And this is usually stuff that's used in the industry, because you're not going to wait to boil the sample and do that. We actually want rapid analysis. And then lastly, we have what we call your benchtop rapid analyzers for salt. And we have your salometer. When we talk about saline, we usually talk about salt solution. So the salometer uses hydrometry. It measures the brine strength. Brine is obviously your salt solution on a scale that indicates the percentage salt saturation. And then you also have the salt refractometer that you guys have used somewhere in Food Technology 1. And that one works similar to measuring of bricks in sugar. So you just take a drop of your salt solution, you put it in the refractometer, and then it tells you there's 10% salt concentration. Easy peasy. However, it won't tell you the type of salt, because there's different types of salts out there. Potassium is a salt, sodium is also a salt, but it will just tell you the concentration, so it does not quantify. The quantification will be then when you're using your ion selective electrodes or you're using your Vollhardt method. So this recording is going to stop here. Thank you very much.